Hi, I'm Chiu from Kwok-Fai Cells Lab. Today I'm going to introduce you how to do the intravitreous injection so that we can produce animal model or prevent ocular disease progression. Here is the schematic image to show the location of injection needle in the red eye. The needle is punctured into the posterior chamber at a 45-degree to the scleral surface. Using this 45-degree angle, we can avoid injury of the lens during intravitreous injection. We'll do the intraocular injection under the operation microscope. The rat will be anesthetized by ketamine and salicin cocktail. Local anesthesia and antibiotic ointment will also be used. We use glass pipette to do the injection. We are using pipette puller to prepare glass pipette. Then we need to get rid of the very sharp end of the glass pipette. We can connect it with a 1 ml syringe.